the topic of the lecture of the lessons was actually when and why using micronuclei in study. I'm generally using micronuclei in humans, but I did also some example in animals, in toxicological studies. And so one of the questions was why, what is the piece of information which is given to you by measuring micronuclei? And you can use it to measure exposure, and this is an important issue. You can use micronuclei as an early biological effect biomarker, which is, I would say, which is the most common definition of micronuclei, because the idea is that you have an exposure and you see the effect, the early event, the effect in the human body or the animal body of these exposures. I mean, there is a number, and Elga talked very well about, I mean, DNA damage, especially genomic instability, and there's a number of events. You measure that, use micronuclei to measure these events. I don't know if you remember my previous lessons in which there was a street going from exposure to the disease, and you have a number of milestones. Maybe DNA damage, genomic instability, inflammation, and micronuclei is really important because it helps you to evaluate some of these milestones. And you can use it also as a susceptibility biomarker. It's not a genetic susceptibility, but it's, I mean, if you have a genomic instability, you can consider this condition as a susceptibility to further to other damages. I won't talk about that. And then what is really important for epidemiologists, you can use biomarkers like the micronuclei and validate them as a risk biomarker. That is, obviously, we are most of the time in these courses talking about the link between exposure and micronuclei frequency as a marker of exposure in early events. But what is really important in this biomarker that you can estimate, you can try to validate what's the second part of the path. So is there a link in between the frequency of micronuclei, the presence of micronuclei, and the risk of disease, cancer or other diseases? Edgar presented the conclusion for the German-speaking group of mutagenesis, that they concluded that there is evidence, experimental evidence linking the genomic instability, the frequency of micronuclei to malignant cancer, but there is also epidemiological evidence, and I will briefly talk about that. And okay, in a way, that was already presented in the previous presentation. Actually, there are a number of endpoints that you can measure, because one of the critical points for using micronuclei is that you can evaluate a number of different effects. It's an early biological effect with the S, is a plural, because we have a number of different events. And this is the assay done in vivo in lymphocytes. And obviously we can measure apoptosis, necrosis, gene amplification, the presence of dicentric chromosomes, and or chromosome breaks or aneuploidy. I mean, when we have chromosome loss. So that's really, with just one assay, you can measure a number of different events. And another important issue is that there is a mechanistic basis to justify the link between what we observe when we measure micronuclei and the mechanism leading to a disease. And this is valid for different pathways. For instance, the typical pathway was for diseases, that's this one, the last one, when you have rearrangements in the chromosomes within the nucleus, which you have aneuploidy or you have chromosome breaks. That means when you have a break, chromosomes tends to rearrange. And if there is a reciprocal translocation, in many cases you have gene amplification or tumor suppressor gene deletion and so on. This is, we know that cancer, atherosclerosis, and there is a number of diseases. But in our day, there is a lot of diseases which are driven by inflammation. And actually, it was anticipated also in the previous talk that there was some events like the fragmentation of micronuclei that in a way activate a number of pathways. And a good example is the CDAS-STING, which is an important pathway leading to inflammation, and the activation of the cytokine pulse in a way. And so in a way, we have basis to link the frequency of micronuclei to a number of diseases that may be induced by mutation, but also by other pathways. So that means cancer, cardiovascular diseases, neurological diseases, disorders, or whatever. So we can try to use it for several purposes. And however, one of the example is, you remember the first box was when we want to use micronuclei as a marker of exposure. And you know, for instance, there is a great interest in dosimetry in when you have to measure exposure to ionizing radiation. And for instance, when you want to do that, and this is important because you have, there is one person that was exposed to an accident, for instance, or is occupationally exposed to ionizing radiation. So you want to know what was the dose that he received or she received, and you don't know. So you measure the damages in the chromosomes, and you can estimate the dose. Generally, this is done measuring dicentric chromosomes, which is a measure of chromosomal aberrations. But it's very tedious, really. You have to score several hundreds cells showing metaphases, which is very long time. And micronuclei can do the very similar results in a much easier way. Here I just presented an example in which there was you see how linear is the dose. In this case is nearly linear, maybe not linear as this, but I wanted to say that this is an important application of micronuclei, that you can really, in this case, measure exposure. You are more interested in exposure than in the effects. And so as a marker of early events, I present you a couple of examples. One is in fishes, and the other is in humans. And this is a study done in, I mean, there was a typical important public health damage, public health risk, like what happened in Fukushima. There was nuclear power plant, there was this accident, and in a way there was a dispersion of ionizing radiation in a very large area. So the idea was to try to estimate how was the impact of the nuclear power plant trash into the environment. And which is really important because, okay, you can measure in humans, but maybe that animals living there, so they're completely exposed and 24 hours per day exposed, maybe a better marker of that. And this is what pushed these researchers in Japan to do that. And they worked on a kind of fish, which is medaka, the wild medaka in Japanese pronunciation. And however, they found two different habitats for these fishes, the wild fishes. One was exposed to, okay, one was pretty close to the nuclear power plant, and the ambient dose, the equivalent rate was, which is expressed in microsievert per hour, is like that. Another is in a way located in different directions toward the plant, and the microsievert per hour was maybe 10 times higher. And so there was a good condition because there were two reservoirs of the same type of fishes, and so they did the analysis. And I'm presenting a little more detail this example because there's some overlapping with the possible problems that you are called to address or to solve, not to solve, but at least to try to solve, to try to address. In this case, for instance, you see that there was a classical approach is drafting circles around. Circles which identify area with progressive distance from the point of exposure. This can be done, obviously, if there is a point and the exposure comes from a single point. That's much easier. So you can measure the distance and associate the damage to the distance. In this case, for instance, was done, you see that there is a number of circles with different colors, and the two reservoirs are shown here. Here's the nuclear power plant, and you see that the radiation are not homogeneous. There are, in this case, distance may be a good marker. You see that that points which are far away, the nuclear power plant, are better, but it's not. I mean, in this case, distance is not perfect because there are sort of part northern are much less exposed that area in the southern part. So obviously the hills, you have to pay attention to the presence of wind, the prevalent winds, and so on. And so they measure, they put some dosimeters in the environment, and dosimeters are some rod like that in which they put it at different point in the ponds, in the lake where fishes live. And I gave a little more detail because I want to just pay attention that when you design a study like this, especially in the wild animals, you have to pay attention to a number of things. Obviously, the habitat is one of the issues. And for instance, the wild medata has a number of different genetic subtypes. So you must be sure that you compare peer with peer and not peer with apples. And this is one point. For instance, they use also some blinded dosimeters just to be sure that to have some controls. And that was quite an interesting, well designed and studied. And they had also some Aquarius medata, and that they use with known doses. And this is quite important because they measure in micronuclei frequency. You see, in cultivated fishes, they knew, so there was a kind of dosimetry based on that. So they knew what to expect from the wild, for the wilderness. Their study at the end of the story was completely negative. You see that this is the mean of micronuclei frequency in medata in the point 1 that was less exposed, and then the point 2, the habitat number 2, which was very highly exposed, 10 times more. But that's the level of this microsievert per hour, 10 microsievert per hour was not enough to remove the micronuclei. So in a way, you can size the risk for the population, the risk for the environment based on that. Okay, that was another example is based on humans here. And I'm talking about urban air pollution, which is another very controversial issue because it's controversial for several reasons, because it's difficult to assess exposure. It's difficult because people living in urban areas has a particular lifestyle. I mean, they smoke more than people living in the countryside. They eat uh, more trash food than people living outside cities. They are more stressful and so on. So there is a urban factor that you have to try to exclude to understand if there is a real effect of air pollution on genomic stability and genome health. And so I'm presenting the result of a meta-analysis in this case, because in a way, meta-analysis is a very powerful epidemiological tool that when you have a number of studies that none of the study has enough strength, enough number to be conclusive. I mean, you can try to put all together the studies with a reliable methodology, with that were good as a study, but not good enough, not strong enough to be conclusive. You can put it together and calculate because systematic review provide you a qualitative information. But from meta-analysis, you can just get quantitative measurement. In this case, when I say quantitative, I say frequency ratio or mean ratio, which is the ratio in between the micronuclei frequency in exposed population to the micronuclei frequency in control population. So, and the ratio between these two measures is like a relative risk. If it is one, for instance, it means that the mean are very much the same. If there is more than one, it means that exposed people has much higher micronuclear frequency. And that's what has been done. You see here that there was a number of studies investigating micronuclei in exposed population. And there is one difference. I'm introducing one difference, which is the different tissue that you can evaluate. I mean, before Elda presented very well a number of different systems, states that can be done. When you work in humans, okay, you can use other systems, but the two more popular systems are measuring the micronuclei in lymphocytes or in exfoliated buccal cells. And this meta-analysis was done in both systems. And you see that choosing the tissue may be an important choice because you have to choose the most appropriate tissue. In this case, we did the analysis. In this case, are exfoliated buccal cells. You see, this is a typical forest plot, it's called, of the meta-analysis. The line like this is the line of frequency ratio equal to 1. That means that there is no difference in between exposed and controls. And you see here that almost all studies had a significant, but two had a significant increase in micronuclei frequency in exposed population. And overall, you see, this is a typical, again, forest plot for meta-analysis. We have to go there where there is the diamond sign, which reflects the overall estimation of the risk, which is 3.16. That means that generally the summary results is that people exposed to urban air pollution has more than 3 times higher frequency of micronuclei. So that means something. And there are also studies done on lymphocytes, and you see that there are many negative studies. Some are not completely significant, and just a few were significant. The overall estimation is 1.29. Is still significant because you see there are many studies, large populations, but remember, to be significant is not always so important. What is really important is the amount of the risk. In this case, you have an increase of 29%, 30% in the frequency of micronuclei, which is significant, which is important, but it's not. That means that lymphocytes probably are not the best tissue to investigate in an exposure that you can inhale, and for respiratory issues, much better in this case to investigate buccal exfoliated cells. And talking about, if there are any questions, you can just raise your hand and I'll try to answer. And I understand this is much different from the previous presentation. And talking about different tissues, I mean, I wanted just to tell you something about because it's really important to select the right tissue to sample. For instance, talking about the lymphocytes or micronuclei, in a few years ago, that was 2011, I believe. Yes, 2010. So a lot of years ago, we did this study and we compared a few studies measuring the same group micronuclei in exfoliated cells, epithelial cells, and in lymphocytes. And the results was pretty interesting. I mean, the mean ratio estimated for exposed with lymphocytes or with buccal cells are pretty much correlated. And so we were happy with that. But now, as a international collaborative group of micronuclei, we were considering that, okay, but exfoliated cells has a number of advantages. You can take the samples from children. Consider also this, if you are planning a study in an exposed general population, and I mean, you have a number of ethical constraints. I mean, you cannot take, it's difficult to take blood from children, for instance, if you don't have a real purpose. And the research is often not considered enough. Taking exfoliated cells is much easier. And the parents, they give you the cells of the children, but not the blood. And the same for people with mental disability, for instance, or people in which it may be difficult to get consensus. So, and furthermore, lymphocytes, you need fresh blood. And you have to cultivate cell for 2 hours. So it means that you have to come in the lab on Sunday to add cytoplasm, which is a problem. Whereas you can keep exfoliated cells in solution even for a couple of months. So you can do field studies more easily. So what we did with this friend, Naz Muller, not Franz Muller, and we reviewed the literature recently, and we understand and we found 130 studies in which they measured micronuclei in both tissues. And so we are able to repeat the same analysis, and the strong correlation is confirmed. And interestingly, this association, this correlation, sorry, is not the same. It depends upon the study. Because if you do a study on occupational environmental studies, like the one we saw before, the correlation is good, but it's not that good. It's 0.4. I mean, but if you go to cross-sectional studies in which you investigate diet or lifestyle or intervention studies in which, I mean, exfoliated cells and lymphocytes may be not that different. So the correlation is 0.9, 0.94, 0.92. That means that you measure essentially the same damage, even if you use very different tissues, which is very good, which is interesting. That means you have to pay a lot of attention because you may select one tissue or another depending upon the kind of exposure and the kind of population that you want to investigate. And okay, I will skip this part. I was talking about COPD and lung cancer, which are extremely associated with micronuclei frequency. But that's the point. I want also to spend the last minute of this talk saying that micronuclei is something which is a kind of something, is a biomarker which is that raise a lot of interest, not only because it's a good marker of exposure, good marker of intermediate steps towards diseases, but also because it's associated with the presence of diseases. So this the condition of risk biomarker is something really important because it makes when you have an increased frequency of micronuclei, that means that that person, that animal have an increased risk of disease. So that push a lot the relevance for public health. So if you find a population with a higher level of micronuclei or a species or whatever, that means that a real risk for the health of that population. That makes a difference. And these are the results of a special issue that was dedicated a couple of years ago, and I was also part of that, and in which we try to review the existing literature linking micronuclei frequencies to different diseases, to say so how many diseases are linked to micronuclei frequency and what was the strength of this association. And actually, results were very interesting. This is the paper summarizing the results of the special issue. And you see that mean ratio is exactly the same. In this case, frequency of micronuclei in people with the disease versus people without the disease versus control. And you see that there is one is the line of no effect. We have like blood cancer, leukemia has like 5 times higher micronuclei, or head and neck twice, breast cancer, lung cancer twice, and so on, or also colorectal cancer, and even inflammatory bowel disease have a higher frequency of micronuclei. This frequency was not present only for, this increase was not only present for cancer, but was present as also in other chronic diseases like cardiovascular diseases, diabetes. People with diabetes has like 4 times higher frequency of micronuclei. There's a very interesting paper on glycation and the association with inflammation and genomic instability, or infertility, and so on. There is a number of conditions. Obviously, micronucleation generates genomic instability, which is a very general effect on the cell. And that means that may have an impact on several pathways. And this is the epidemiological proof that this is actually like that. Please.
What the error bars, what does it represent? Is it the normal standard deviation? Is it the standard error? Because they are, for example, with diabetes, it's a big error, not? So
yeah, that's the standard error, and because it's a sample, and obviously the differences, it depends upon the number of studies. So in a way, diabetes, there were less studies than, for example, on obesity or overweight, or and actually, or that's the answer. And however, all of this study showed significant increases over the. There is different degree of robustness in the evidence. This is just to give an idea. When you see small bars, variability bars, it means the data are more robust. Statistically, Then there is a epidemiological evaluation. And I was saying that you can measure micronuclei not only in exfoliated buccal cells or lymphocytes, but also in cervix, urine. You can take urine and select, find exfoliated cell, urethral cells, and measure micronuclei in that. And this may be very well associated with bladder cancer. Normally, you see that there was 10 times higher, or cervix and so on. That was to say, is really important. The final two or three slides to say all the results I've shown you linking micronuclei to the disease have a general problem. What's the problem with those data linking micronuclei to diseases? Is a disease that caused the DNA damage or genomic instability, or is actually what we plan to be is the genomic instability, DNA damage that predict the disease? The only way to measure that is there are different ways, but we need mechanistic evidence, but we need also epidemiological evidence to prove it. And so the epidemiological approach is to design a cohort study, to design a prospective study. The idea of prospective studies is that you have all people which are healthy at the moment when you start the study. You measure micronuclei in these people and you follow them up. And you count how many people develop the disease among those with very high level of micronuclei at the beginning versus those with low level of micronuclei at the beginning, when all of them were healthy. That's the approach. And conceptually, you generally have exposure, you have a biological event that may be a chromosomal rearrangement, symmetrical translocation, then you have a disease. We cannot measure actually the target event in the target cells. So you have to use surrogate marker like chromosomal aberrations or micronucleation. And what we really have to demonstrate is that these unstable intermediate steps are linked to the cancer, to the disease. And to do that, for instance, in the past, we did a study, a cohort study. It was a particular study design, which is called historical cohort study, because you selected people, studies done long ago, like you started that time. And the present is like it is the future. I mean, there was a follow-up lasting for several years. That was based on a large, relatively large group of people, and the results in the end were showing that people with the lowest level of micronuclei had a better probability of surviving, or the lower frequency of cancer. In that case was on cancer. And obviously, this was the start of public health application, because obviously you need to make more, to strengthen this evidence. And for instance, last year we published together with Mike Fennett a paper just to try to figure out what are the steps to validate actually this biomarker, for instance, for individual risk assessment. So you can say, you have a high level of micronuclei, I can make a personal assessment, risk assessment. And obviously there is a number of steps. More or less we are at this point. We need now to have more prospective studies, and we have to do clinical trials. That's the point. For instance, to say you have a certain level of micronuclei, I can change your level, I can lower your level, and then your risk is changed. That's the challenge for the next year to come. And the last, I believe, yes, is the last to say there is a number of examples, a few examples in which micronuclei can be used in clinical practice, which is there are not that many, just to see spleen function or to see if you have a vitamins deficiency. I mean, monitoring the dosimetry, and that are, I mean, they are not, not very important application. Much more important, the one in the middle, the yellow, in which we don't have enough data. We have promising data to estimate the risk of accelerated aging, chromosome frailty, or cancer, or whatever. And in this case, more data are required before we can really establish this link. And there are other in which the evidence is really inadequate. We need more data.